To predict which genes will be expressed after single gene induction, this paper uses transcriptional model in yeast cell experiments. So this paper is called Learning Causal Networks using Inducible Transcription Factors and Transcriptome-wide Time Series. It's from Hackett, Paltz, Coram, Vranic, Kim, Baker, Fan, Hendrickson, Brendel, and McIsaac. It's from, I think it's from 2020, yes, 2020, partially funded by Google Research, but also Calico Life Sciences. Both sort of California. So in this paper they did both an actual physical experiment which produced a dataset, and at the same time they also modeled the behavior using whole cell transcription model. Yes, so in the abstract they mentioned that dataset constructed they constructed a dataset by independently inducing hundreds of transcriptional factors and measuring time courses of the resulting gene expression responses in the budding yeast. They had a whole cell transcription. They created a whole cell transcription model, which they used to predict and validate multiple new underappreciated transcriptional regulators. They also produced the magnitude coefficients in this model and shown that they are predictive of genetic interaction profile similarities. So direct and indirect molecular interactions that achieve particular state can be described as regulatory edges that collectively form gene regulatory networks. So, this influence of expression of one gene to expression of other genes can be modeled as a graph, and they call this, or network, and they call this a gene regulatory network. And that's what they sort of try to contribute to, that's what they try to model physically, and from the model they try to extract sort of these coefficients in the graph in this network, because one gene may have bigger effect on another gene, induce transcription of the other gene stronger, with more powerfully. At the same time, one gene can also reduce transcription of other gene. To actually measure this, they had to use indirect methods. So instead of measuring explicitly the transcription of the DNA, I think they measured just the RNA transcription, which is a secondary effect, but much easier to measure. And they did multiple time steps measurements to be able to model this easily. So what they measured is they had some original sample, they had some RNA, they had some RNA proteins in there, they had some counts. Then they activated one gene and measured the counts of different RNAs over time, which they used as the input to a model to model this gene regulatory network. So what you see here, that activating one network element followed by dynamic measurement of all other network elements. 200 induction experiments in which single yeast transcription factor was rapidly induced from a beta estradiol responsive synthetic promoter, and full transcriptome differential expression was subsequently tracked, typically across 8 time points. So they have a diagram here of how their experiment actually works. So in each experiment, one transcriptional regulator, which an inducible promoter, is rapidly overexpressed in response to this beta estradiol introduction. Example of 3 genes which they measured as responding with different kinetics following the induction of gene A. So they get some results, and they struggle what activates what, right? They have 3 genes activated, overexpressed. They measure them in the sample with some responses, but they don't know if there are some relationship, if there's some sort of cascade. So how they find out? So they find out by doing more experiments, activating the other genes and then observing. And by that they can, and also observing in time. So they did 200 experiments of these. Just to mention, of course, these counts, these relative counts of RNA instances in the sample cannot go to infinity. So when you have the model, you have to model in such a way that over some time the derivation, the increase should be zero again. So you start, somewhere low and you end up somewhere high or somewhere low as well. In the case of an inhibitory interaction. So the model is constructed as a set of gene-level regression models that predict the rate of change of each target gene as a sparse linear combination of all genes' expressions. So they have this sparse linear combination of our expressions, where all the y's here are actually relative counts to the control strain at the time 0 of a gene I in experiment J at the time t. And so the first part is the effect of all linear effect of all other genes, and the second term here is, I think, this effect that prevents going to infinity, sort of the dampening effect that after some time you need to get a sigmoid. So that's why you have this other part, which also depends on the target transcript count. So each regression model predicts the rate of change of target gene based on other regulators. So what they model here, this linear equation on the right, is with this they model change of the count in time, in one time step. So each regression model predicts the rate of change of target gene based on other regulators. These instantaneous estimates can be integrated to provide estimates of log2 fold changes. Grossly, the model explains 43% of variability in log2 fold changes. While the model does not account for all expression variability, both dependent and independent variables are directly determined by the experimental data. Accordingly, our inability to predict one gene's regulation does not immediately affect the modeling behavior of all other genes. Yes, so one thing I haven't mentioned, there is a logarithm here as well. That's because otherwise you want to model linear effects. But these interactions would lead to. Yeah, so this is sort of way how to linearize the equation. So the discussion. First, only a small number of genes in the previous data set that were marked as they are transcription factor bound, so they are part of this network, actually responded in the typical experiment. And most of the responses they measured were actually undescribed. So we can see that there's a lot of unknowns in this area. The data sets are not accurate just yet. By integrating hundreds of time courses with the dynamic system model that explicitly includes time, we can make predictions of new regulatory interactions without use of prior knowledge. When fitting a genome scale regression model, we made a number of assumptions about the biological processes to include versus those we should ignore based on our experimental design. in doing so, our model may fail to capture a number of important regulatory phenomena, including complex combinatorial regulation, post-transcriptional regulation, post-translational regulation, localization, and regulation due to non-proteins. So I hope you got the spice of the paper, the juice. And yeah, I hope to see you next time. Please unsubscribe, dislike, and delete comments.